Okay, so this has become my kind of passionate subject for the last year or so, and I'm very glad that uh, the lab was willing to give me some time to talk about this because I feel it's one of those things that has gotten lost in the shuffle of complexity, methylation and glutathione issues. And so I was struck by this lecture I heard from our leading representative in functional medicine, Dr. Mark Hyman. And there's even a representation of this on his blog post. If you want to look at this later, you can Google Mark Hyman methylation and glutathione, and it'll pop up. But in the talk, he mentions the two most important processes in the human body, period. The things that keep us alive. And number one is glutathione. Number two is methylation. In fact, I think he has number one methylation, number two glutathione. So these are not ancillary things that we should check in chronically ill people. These are not things that you just kind of do if the person has an MTHFR defect or you suspect a SNP. These are fundamental body processes at the highest levels of biochemistry, cell physiology, human physiology that have to be addressed for a 5-year-old kid that's autistic, for a 25-year-old person that's depressed, for a woman that wants to get pregnant, for a guy who just wants to ride his bike faster on the weekends, for an executive, for a chronically ill patient, for anybody who's walking around in a human body. These are the two fundamental factors that we have to check and make sure that they're working. And so somehow they both kind of been relegated to these obscure, complex, chronically ill situations. But really, when you look at it from the biochemistry viewpoint of a brilliant scientist like Mark Hyman, or from a brilliant scientist like my current teacher, Dr. Richard Lord, and you're just thinking from that 30,000-foot view, these are two processes which, if they're not working well, are going to lead to human death and misery faster than anything else that could go wrong in the body, right? So this is just part of my general workup on every patient now. Again, it could be a 15-year-old kid who just wants to do better in school. It could be a 45-year-old with fibromyalgia and chronic fatigue. And so from the biggest picture, I'm thinking again, what is methylation? What are we methylating, right? And the vast majority of methylation resources are directed towards creatine production, right? And membrane production. So you can see right away, okay, we're doing energy stuff for the muscles, we're making cell membranes with the phospholipids and all that, right? And then smaller percentages of the methylation processes go towards the things that we're more familiar with, right? We're methylating DNA, we're dealing with hormones, dealing with neurotransmitters, et cetera. And then glutathione being the master antioxidant that we can control, millisecond to millisecond in terms of protecting our tissues. So those are the kind of the big picture issues I want to get into today. Okay. Oh, and then supposed to mention, we have a boot camp coming up. This is like our most popular boot camp. Go figure. It's on telehealth business essentials. It's basically like a practice building, business building workshop, and it's 2 months long. We get into financial planning, business planning, strategy. You just kind of force the doctors to deal with their business and help their business grow. So every year we do this at least once. It's been our most popular one for a bunch of years in a row. And if you feel like tuning up your business and spending a couple months in the summer doing that, then you can join us for that. And you get a big discount with the doctor's data discount code there too, okay? All right, so back to the subject matter here. We want to think about how methylation and glutathione are connected and why we have to test and treat them together. That's a pretty important one. And why some people that will react poorly to glutathione-related products need to methylate better, why some people react horribly to methylation support because their glutathione's not working well, try to unravel that whole thing. And then talk about all the supplements that are involved, including the two sort of sleeper products, betaine and choline. And betaine and choline, I think for most of us, should just be part of a comprehensive methylation support program. And that may not be so obvious from every lab test, but those are products that you just want to keep in the front of your mind, even if you don't have lab data indicating that the person needs them, as general support and background products to make sure your other methylation support works well. And then, of course, thinking about B vitamins, how they overlap and how they play into methylation and glutathione production, and how mitochondrial problems and oxidative stress can be part of this whole methylation conundrum. Okay, and then the big picture supplements we need to learn how to use: folate, B6, B12, and then B complex in general. And the general rule there, you probably already know, but if you're going to give a B vitamin, you always want to start with B complex and then add to that the extra specific B vitamin that you want to boost up. So B complex as a base layer, if they need more folate, you give extra folate on top of that. And then methionine, and we'll look at some of the other amino acids too that are involved in methylation with a single carbon pool, but methionine is probably the most well-known. And then for glutathione support, obviously you can use glutathione if you're into that. Super great product. A lot of companies have liposomal glutathione, all kinds of fancy delivery systems now for glutathione. There's even some glutathione capsules that make it through the gut absorption problem that we used to have with glutathione. So advances in glutathione support are quite amazing, and there's all kinds of products out there for that. And then you can also go the old-fashioned, old-school way for people with low glutathione and use NAC, also known as N-acetylcysteine, or some people prefer glycine, glutamine, the 3 amino acids from which we make glutathione. Most often people use NAC. And one of the things about NAC or N-acetylcysteine that I found is that if you underdose people on it, it doesn't work that well. Shocking. But, I found most of my career I was doing that. And so when I started to work with Dr. Laura, I was like, well, what should the dosages be? And we started to look at it and we're like, oh, okay, well, I guess I'm a little bit off on that. And so starting dosages, 3,000 milligrams a day for N-acetylcysteine. If that's not helping the person after a few weeks, going up to 6,000 milligrams. I know that sounds like a lot. It is a lot. But we need a lot of, some people need a lot of glutathione support. So you don't want to underdose people on NAC. I have patients coming in literally every week, but I've been on N-acetylcysteine for 3 years. How much are you taking? I take 500 milligrams a day. And I'm like, well, we're going to start you at 6 times that amount, and we're going to go up from there. So oftentimes the failure of the program can be dose dependent, and we don't want to overdose people. You certainly don't want to harm anyone, but underdosing them is not great either. And if you're nervous about it, put them on the program, 3,000 to 6,000 milligrams a day, and then stop after 90 days and retest. We're not doing this for a lifetime. So the higher that you're going to dose a person, the more often you should retest. And 90 days of a really strong N-acetylcysteine program should boost anyone's glutathione up. And if that's not happening, then you're still underdosing them, and then bring it up a little more. So use the testing to cover yourself. Be more aggressive with your dosing and retest a lot. That's what the old school naturopaths did, so don't shy away from that, because you don't want to have someone come, go see some other doctor 3 years from now, having been on N-acetylcysteine at 1,000 a day, and it just never moved the needle, right? And then, of course, I mentioned choline and betaine already. Okay, so that's like the ballpark of what we want to learn how to work with. And then I think the thing that's been shocking to me about understanding methylation and understanding glutathione is how amino acids and the amino acids roles in protein synthesis tie into all this. And so I think it's worth taking a moment and thinking about the protein synthesis and then non-protein functions of amino acids. Because in my experience in our field, we all focus on the non-protein functions of amino acids almost exclusively, to the extent that even if you ask a doctor point blank a protein synthesis question, they rarely get it right. So I'll give this one away, and if you guys ever take one of my other classes, you'll get this one right. But the question is, so what's the main purpose of tryptophan in the human body? What's the number one thing that tryptophan does? That's the most important thing. Every biochemist agrees on this. There's the International Journal of Tryptophan Research, believe it or not, that's like a real journal. They all agree on this. This is not like something that people debate. There's an instant and accurate answer. What's the most important role of tryptophan? If you look it up in any biochemistry textbook, it'll say, most important role of tryptophan in the human body is protein synthesis. All proteins require tryptophan. And then the next sentence will usually say, and to a lesser degree, in a very small percent, tryptophan is also involved in ... serotonin production, melatonin production, etc. Okay, so these individual amino acids, these 20 amino acids that we use, their primary role is in protein synthesis. Their secondary roles are what we usually focus on. And so when we're thinking about enzymes and why SNPs even happen, we want to think about protein synthesis as the core of the problem. Okay, and these enzymes could be cytochrome P450 enzymes for detox. They could be pyruvate dehydrogenase enzymes for making acetyl-CoA from glucose. They could be succinate dehydrogenase enzyme for running complex 2 in the electron transport chain. It could be MTHFR as an enzyme. It could be any of these enzymes that we're talking about. And so we just want to take a few minutes and talk about how this works so you have like a really clear conceptual framework to explain this to patients with. And I think it helps patients a lot to understand this. So I always try to think about patient education. And so what we're going to get into just for a second is protein folding, right? Protein folding. So here we are. We have these amino acids, right? And your body takes them and strings them together using something called DNA. That's the purpose of DNA, right? Is to have a code, like a secret little code that the genome is, that tells the body exactly what sequence these amino acids should be in. And then once that all gets done, which is a miracle that that can happen, then those amino acids, because of the sequence that they're in, form structures like alpha helixes, which to me looks like kind of like a spiral staircase kind of thing, or a beta sheet, there's sort of more of a sheet. Depending again on the charges of the amino acids and how they're aligning with one another, they then fold up into these tertiary structures, and then the final protein structure is made based on the sequence of these amino acids. If one amino acid is out of order, and this can happen with an enzyme. I mean, just think, if you were your DNA and you're trying to put together an enzyme that has like 500 amino acids, you'd mess up once in a while, right? And there'd be like one amino acid out of sequence. That's a SNP. So then when this thing goes to fold, because the folding process is based on the sequence of amino acids and their charges and how they attract and repel one another, if one amino acid is slightly out of where it's supposed to be, is swapped out for another one, the thing's not going to fold right. And that's basically the structural defect that we call a single nucleotide polymorphism, right? There's a structure with a morphology problem. And when that happens, then it's really hard for the coenzyme, in this case, B vitamins, to slot in and fit into that enzyme properly. Okay? And so the enzyme, otherwise perfectly constructed with this one amino acid out of sequence, is going to then require a much larger amount of a given vitamin, like a B vitamin, to make that enzyme work. Okay, and that ends up being a pretty big problem. And here's the typical way that it's depicted. You have an inactive enzyme just sitting there that's been made but hasn't done anything yet. Then a chemical compound comes along, but it can't bind to that enzyme without the coenzyme. Okay, so now we've got the coenzyme, that's the little purple thing there. That's the B vitamin in our example, right? Now we got the B vitamin connected to the enzyme. They're together. And now when that compound comes around, it can connect and gets locked in and bound to the enzyme, right? Now this whole chemical reaction can take place. So, but what we're worried about, especially, is when the enzyme is not folded quite right. And so the coenzyme or the B vitamin has to be provided in large amounts to get this entire process to work. And we have, a large majority of patients that we work with that are going to have some kind of defect along these lines. So again, what is a coenzyme? It's the vitamin, or in some cases there could be a mineral, right? Coenzyme or cofactor that's going to activate and make that enzyme work. And again, these are the non-protein portions of the enzyme. But my gripe about even like the term cofactor or coenzyme is sort of diminutive, right? So it's like if you get on an airplane, which I used to do all the time for work, as Jonathan well knows, I used to go to all these conferences every month. We'd be like, oh, where are we now? Is this Chicago? I don't know. Maybe it's Denver, wherever. We do constantly, but that hasn't happened recently. But anyways, back in the day when you get on these airplanes every week, and you get on a plane, and if you're lucky, there's two people right at the front of the plane. And one is a pilot and one is a co-pilot. And you're pretty clear that the co-pilot is the less important person on the plane. And you're thinking, okay, if someone has a heart attack here, I hope it's not the pilot, because if that happens, then the co-pilot has to fly the plane. So right away, as soon as we say co-pilot, you're thinking that's the less important. It's the same thing with like a co-star in a movie. They're not the star, they're the co-star. So I think that when you learn this stuff in biochemistry classes and you hear coenzyme, you're like, oh yeah, it's coenzyme. Like, it's not as important as the enzyme, just like the co-pilot is not as important. But that's not really true in this case. In this case, the plane can't get off the ground without the coenzyme, okay? Like this whole thing completely is dependent on the coenzyme being there. The enzyme is useless without it, completely useless. It's just sitting, in fact, it's just sitting there, you know, wasting space and time, not being able to do anything. So when we say B vitamin is a coenzyme, it's playing a major role, as major a role in this whole process as the actual enzyme itself is. So that's just important to remember because it's easy to kind of diminish it. Now, here's another enzyme. And again, just beautifully folded. And this one turns out to be incredibly complex. And gosh, look at it. You know, you can imagine how many amino acids are strung together there. And the more complexity, the more number of amino acids, the more potential there is for it to be constructed a little bit wrong, right? And so now we want to talk about, that was MTHFR, by the way, in case you didn't get that reference there. So now we want to talk about the mechanisms, right, that make up methylation. And then we're going to tie this later to the mechanisms that control glutathione production. And then we're going to look at some lab examples so you guys can do these tests. And again, this is not like an optional test. This is like you should do it on everybody kind of test. Can they methylate? Are they able to properly handle DNA, neurotransmitter levels, hormone production, detoxification pathways? Are they methylating? Can they make enough creatine? Can they make cell membranes properly? These are, you know, kind of mission critical processes for everyone that we work with, whether they are chronically ill or not. And then there's some basic, you know, I don't know what you call them, mechanisms or processes that we want to make sure that we think through without making it overly complicated, right? And so within this process, like within all these other processes, everywhere that there's an enzyme, there's a potential for the enzyme to not have been formed properly, right? To have an amino acid sequence problem or a SNP, which means that that enzyme's not going to perform that great. And so when that B vitamin tries to connect with that enzyme, because there's a slight alteration in the shape of the enzyme or a SNP, more of that B vitamin is going to need to be present to make the chemical reactions work. And turns out, going back to Roger Williams in the 1950s, if you haven't read his book, it's a must read, Biochemical Individuality. Dr. Roger Williams was written originally in the 50s and updated in later years. But he was saying back then, as he was discovering the B vitamins, he's one of the scientists, I think he discovered 3 of the B vitamins. As he was discovering the B vitamins, he was realizing that animals, including us, including humans, had huge genetic variations in their need for individual B vitamins. Okay, Dr. Williams put that together in the mid-1950s. That was a long time ago. And in fact, if you look at his original writings, he even talked about mental health problems like depression and different the mental health disorders, some pretty serious ones indeed, that were related to these genetic variations in B vitamin needs. It's fascinating. Okay, this has been going on, I don't know, that was like 60, 70 years ago, right? The mid-1950s. This is not a new idea. Now we can actually measure the genes, measure the SNPs. We have these amazing labs where you can measure all the functional markers that you need to start to put this together. Now, the main factors then you want to think about from again, I'm trying to look at the biggest picture here as possible, is that are there, you know, these variables, how can you say it? Like, what are the main variables that are driving this, right? And to not just get into the minutia of the problem, but to take a big step back and say, okay, what does the cell need to do to survive? The cell needs to protect itself to survive. And what is the number one threat to the cell? The number one threat to the cell is that it could die. And how does the cell prioritize and deal with its own survival versus its need to help facilitate the methylation process, okay? And for that, we have an explanation that the scientists have handed us, which is that it turns out that this struggle between methylation and being able to have the nutrients required for methylation and glutathione is something that goes on in the body moment to moment, day to day, hour to hour, minute to minute, second to second, all the time. Okay, so we're going to talk about this bigger picture. You just want to keep that in the back of your mind. So, and then on the front lines here, we're looking at B vitamins and amino acids. And then Well, last time I checked, right, DNA synthesis requires folate and amino acids. Protein synthesis that we talked about a moment ago requires B12 and folate and amino acids. Your metabolism absolutely is based on amino acids and B vitamins, right? Your neurotransmitter production requires amino acids, but they don't work at all without B vitamins. Hormone production absolutely requires amino acids and B vitamins. In fact, sometimes when I sit down and look at the supplement programs I design for patients, I'll look at a whole complicated program. It took me like an hour and a half to put together. And I really added up and like, wow, this is all like amino acids and B vitamins. This is all that this person needs. Not that it's all that they needed, but like after all that work, it's really just coming down. These are really like baseline important things that you just use over and over and over again. And then I said this a while ago, but I'm saying it again because everyone has forgotten this, which is protein synthesis. What proteins? All proteins in the body require those 20 amino acids, those 20 basic amino acids. And so if you're missing one amino acid out of the 20, you're not going to be able to synthesize proteins properly. What proteins? All proteins, except for collagen, require those basic 20 amino acids. So if one amino acid like methionine is missing, is low on a test, that's going to really not be a good thing for methylation. But that's also going to directly impact the body's ability to synthesize all proteins. That includes every enzyme, that includes hemoglobin, that includes some hormones like insulin, that includes your everything to do with your cardiovascular system, right? The proteins are basically almost your entire body. And then we come back around to kind of loop around to today's topic. We're thinking, oh, well, methylation, like all these other things, requires amino acids and B vitamins together in some sort of synchrony to make it work. And I guess it's also important to just think, what are we actually doing with methylation? Because with methylation, again, I immediately go to chronically ill people and all these obscure things. But the bulk of methylation goes towards making basic stuff: creatine for energy, for muscle energy, and choline and cell membrane production. So, we don't want to always just think about the complex. You also think about everybody needs cell membranes, everybody needs muscle energy, And then I also think that, because there's so many enzymes and they're so interesting, you can't really have time to talk about them all. But this one, it seemed like worth mentioning. And just to give some reference points here too, like at least the last time I looked this up, niacin, okay, which just seems like an innocent B vitamin, niacin, is involved in around 400 enzymatic reactions. Some books say 300. Let's say 300 to 400 enzymatic reactions. Thiamine, over 100. So these are like busy vitamins. So if you're low in niacin, that interferes with 300 different enzymes. That's a lot of enzymes to get screwed up. If you're low on thiamine, 100 plus. B12, now of course B vitamins do all kinds of other things besides just helping enzymes work, right? B vitamins are important for other reasons. We're just talking about the enzyme part. But the curious thing, and I don't know, it just makes you wonder, like, why did nature set this up? B12, it's only really active in 2 enzymes, 2. When I read that, I was like, well, how is that possible? And then why is that even possible? And now, of course, B12 does other things, but how come niacin has 300 enzymes that it's related to and B12 has 2? And then you start thinking, well, I don't know. They're pretty important enzymes, right? Here they are. They're right in here. And I think I have pictures of them too, in case you want to see what they look like. Yeah, there's one of them. And beautiful structures again, right? Look how complicated. And there's the other. Beautiful. Methionine synthase and methyl malonyl CoA mutase. Those are the 2 enzymes that B12 acts with. Let me show them one more time. Aren't they beautiful? I don't know. I love these structures. And it's amazing that scientists can actually draw these out for us. That's a whole nother story. It's very, very complicated to take a string of amino acids and then be able to make a picture of what they're going to look like. There's this huge computer breakthrough that happened last year that allows them to do this in a much more efficient manner. But anyways, they're beautiful structures. And again, they're not to just stare at these things and kind of get like a modern art diagram, but the complexity of them is directly related to how messed up they can get, right? Because if one amino acid's out of sequence, methionine synthase may not work. There's a SNP. If one amino acid is out of sequence, this guy doesn't work. Okay? And so this ends up being super important. And if they're not working properly, then the person's going to need more B12 than they would otherwise. Okay, now here's again another view of all this. And I got off on this thing, you know, we've been working with Dr. Lord for a few years now, and he decided that I was ready at some point to hear these talks. And so he just randomly one day, we work together every Monday and Thursday, right? One of the weeks that we're working together, he just said, hey, Dan, I want to lay down a recording about methylation. I was like, okay, that sounds fine. And I'm thinking, I mean, at least I kind of think I know what at least I know what the word means, you know. And by the end of that day, where he went through each one of these different kinds of folates, I was like in shock. I was just on the ground and in shock. And I was like, I know nothing about anything. And then the horrible part is the next week he came back with another lecture and he started to talk about the single carbon pool. And like, I never really even heard of the single carbon pool, Richard. And he's like, well, maybe you should learn about this, Dr. Kaler. This is kind of important for methylation. And then that wasn't enough. Like in the third week, he gave me this whole talk on glutathione, which tied the whole thing together. So anyways, thank you, Dr. Lord. I'm trying to communicate to other people what you taught me right now. But let's start at the bottom here. Single carbon pool, sometimes it's called a one carbon pool. Did you ever wonder where the carbons come from? Because what methylation is moving carbons around, right? Moving methyl groups around. They have to come from somewhere. So origins of single carbons. Guess what? Serine, glycine, histidine, glutamate or glutamine, and tryptophan are the origins of single carbons. Let me say that again. Serine, glycine, histidine, glutamate or glutamine, and tryptophan are the origins of the single carbon pool, which ends up driving this whole methylation process. The last time that I checked, these are amino acids, right? So you need serine, glycine, histidine, glutamine, and tryptophan to get the carbons to form the single carbon pool that's going to be part of this folate trap that's going to drive this whole process. And without those carbons, there's no methylating to be done. So you can have a patient that's low in serine, glycine, histidine, glutamine, or tryptophan, and that's going to make this whole thing not work that well. So again, this amino acid B vitamin connection runs pretty deep. And then of course you need the folate, and we all know that. There's different kinds of folate, which is something that I don't even want to think about. It's emotionally just too much for me to deal with. It's so complicated. I just don't even want to think about it. You can see that he tried to put it all in this diagram here. He's showing you the different kinds of methylhydra, this a folate, this or that thing. But anyways, clinically, I don't think it matters. You just buy the right folate supplement from the right company and you got that covered, okay? And then you've got your B12 circulating here. And we just looked for a second ago at those enzymes that are related to that process, right? And then eventually you get up to the top here, which is what we think about, where you've got homocysteine cycling to methionine, methionine cycling back to homocysteine. And as Jonathan was saying a few minutes ago, it's a cycle. It's just rotating around. It's moving around. There's movement here. And as part of that movement, we generate these methyl groups, and that allows us to methylate and, in a sense, to make things, right? To make hormones, to make neurotransmitters, to make DNA do what it's supposed to do, and just to, you know, keep human life kind of stimulated and working along. But the key point here is that amino acids provide the carbons which are required to feed this methylation system. So we're trying to take bigger and bigger views of this, right? Going back, it's like when you're on Google Maps, and first of all, you're looking at like the house on the street, and then you go back and then you see like the city, and then you hit the wrong button, all of a sudden you see like Australia, where you're like, oh no, wait a minute, I gotta zoom back in, right? So we're kind of zooming out now, okay? And now we're going to zoom out. If you can just stay with me one more time, we're going to zoom out again in a minute, a little further. This is not a zoom out. This is like a zoom sideways here. But I want to just point out that there is a role for choline in all this. There is a role for choline in all this. And that choline, and remember, choline and betaine should be supplements that you keep in mind as you're trying to get this whole thing to work right, okay? And I don't know, that's not that important. We can skip that. There are all these markers. We can skip that too. This is what I want to look at. Here we go. Right, this is the important part. So this is what I want to dwell on this for a while. Okay, so when we have, again, we're going back to that Mark Hyman, a blog post that inspired me so much a couple years ago, right? Where Mark's saying two most important processes in the body that for human survival. These are bottom line things every functional medicine doctor thinks of. First thing we wake up in the morning, this is what we're thinking about. We're thinking about methylation and glutathione. Okay, and then how are they linked is the next question, right? So methylation and glutathione. So number one here, we have methylation up top, and we here have homocysteine converting to methionine, methionine converting to homocysteine. Here's our SAM, right? Here's our SAW. That ratio shows up on the test that doctor's data has too. So you can see a lot of level of detail in here. Here's that betaine that you might want to use along with your choline as a supplement to make sure this whole process is working well. And then down here, we have the production of glutathione. Now, and this part is interesting, is that your cells prioritize glutathione production over methylation because your cells are prioritizing cell survival. Okay, your cells are prioritizing survival. And if there's a lot of oxidative stress hitting the system, your body has to modulate, and it either has to direct more resources towards glutathione production, or it has to direct more resources towards methylation. It has to choose. Okay, so again, this diagram shows oxidative stress is going to decrease transmethylation. It's going to decrease the methylation cycle itself here and increase transsulfuration, which means that you can make more glutathione. So if there's oxidative stress, your body senses that. Rather than letting the cells die, it brings up glutathione production at the expense of methylation, Okay, so you want to measure glutathione. If glutathione is low, that's a higher priority fix, a higher priority correction than correcting methylation. I'm not saying you could do both at the same time, but if you get a patient who's methylating poorly and you crank up methylation and their glutathione is low, you run the risk of making them a lot worse because you're going against everything that the body is designed to do. So let me say that again. You have a patient and you don't realize it, but their glutathione is quite low. You boost up their methylation process. Now what's going to happen is you're forcing them to methylate more, but they want to transsulfurate, right? They want to bring all this resources that they have down to make glutathione, okay? Because the cell survival, the reduction of oxidative stress is more important than methylating. And this is why, or one reason why, you'll see a lot of bad reactions in people who have low glutathione and you try to get their methylation processes going before or instead of working on the glutathione. You can work on them both together. I'm not against doing that. That's probably a good idea for a lot of people. But if you ignore trying to boost their glutathione and you crank up methylation and they have a lot of oxidative stress already, it's just putting the resources in the exact wrong place. Okay, and boosting glutathione production, again, you can give them glutathione supplements. You can give N-acetylcysteine to bring up glutathione. You see cysteine right there. Some people will combine the cysteine or N-acetylcysteine with glycine. Glycine will help boost up glutathione as well. And so does glutamine. So the three amino acids that we use to make glutathione are cysteine, glycine, glutamine. Most common one that most people use is N-acetylcysteine to bring up glutathione. And so that's a priority and needs to be addressed as part of, or sometimes even before you deal with the methylation process because of this basic mechanism. Okay, so moment to moment, your body is sensing the amount of oxidative stress. And there's all kinds of antioxidants that we have. We have C, we have E, we have beta carotene, we have A. We have all these different antioxidants in the body, but your body can't just like on a dime, crank up your vitamin C levels or crank up your vitamin E levels, right? That's just not really possible. But glutathione can be adjusted in a millisecond, you know? Your body can produce huge amounts of glutathione in really short periods of time. So whereas the other antioxidants are very important, CoQ10 is super important for all kinds of reasons, right? But your body, if you look at the pathways for CoQ10, it takes a little while to make CoQ10. It's like, I think there's like 37 enzymes or something. That, I mean, it's amazing that we can make CoQ10 at all. It's just so much work for your body to make CoQ10. You're not going to be able to get that done in a millisecond. But your body can crank out glutathione levels quickly if it has to. So whereas there's a lot of other important antioxidants, it's glutathione that's regulating our moment-to-moment oxidative stress load. And we're either making a lot of it or cutting back on it, or making a lot of it or cutting back on it. And every moment that you make a burst of glutathione, it's going to pull this transsulfuration pathway down, and methylation is going to take a hit. Okay, so you just want to think again, we're trying to look at this bigger picture view, or biggest picture view, when you're fixing methylation, to know the status of the amino acids, right? So you don't have a lack of single carbon contributors to the single carbon pool, and make sure you know those ones, right? And again, they're, in case you already forgot, they're right here. Serine, glycine, histidine, glutamine, and tryptophan, those 5, okay? And then you want to think again, big picture, the status of glutathione, test it, correct it with N-acetylcysteine, glycine, or glutamine, or all 3, or glutathione itself. And then you should have some much more integrated programs where it's making more sense to your body, right? That this stuff is going to work. Oh, and I just threw this in there because I think it's kind of interesting. But if you really want to get into the details of this stuff, it's in Dr. Lord's new book, where he talks about the different folates and how they all work and all that. And the same thing here with the single carbon stuff, okay? I want to have some time to look at labs too before we get done with this, to see. And I don't know what to say about this, but I should at least mention this. But this part here, like, because part of what's happening with the methylation process is we're devoting a huge percentage of methylation, what was it, like 10, 20%, right, to membranes. And I don't know, it just seems like every time you turn around in biochemistry, organic chemistry, human physiology, it's just like membranes. It just takes so much energy for us to produce, maintain, and protect cell membranes. I don't know, whoever invented this system, it's just kind of a time suck. I wish we were like plants. Plants have cell walls. That's got to be easier to make and maintain a cell wall. But a cell membrane, it's just like crazy town, you know? But one of the things that you're going to improve as you improve methylation is, and this is the argument for giving choline, okay, is that you're going to improve cell membrane function, production, maintenance, protection, and all that good stuff. And so you just want to think about choline in the back of your mind when you're doing these programs, and make sure that you're attending to the whole cell membrane issue, okay? All right, now, if you look at these labs, now you can see all the things that we're talking about just kind of laid out right in front of you. And how easy is this? Oh, and then separately, you can also do a glutathione test, red blood cell glutathione test. Why? Because of all the reasons that we just talked about, yeah? And so you're going to want to focus on, if methionine levels are low, for example, that you would want to give methionine. How much? Well, don't just give a gram or 2. You know, it's just not enough. So I think with most of these amino acids, if you see an outright deficiency, you want to start at the 3,000 milligram range. And, you know, you might even need to go up from there. And it's the same with cysteine, N-acetylcysteine, glycine, glutamine. If you're trying to correct these defects with these amino acids, you're going to want to start around the 3,000 milligram range and not be afraid to go up to as much as 6,000 milligrams. How often do you go up to 6? Not that often. I'd say probably, I don't know, once a month, twice a month. I have a patient that's just not responding to the lower dose, so I bring it up. But you should be aware of that. And, you know, think of 3,000 as the opening round in this whole process, you know, as you're getting started. And then, of course, you got to think outside the box too with other things, because if you see an elevation in homocysteine, then you not only know that there's some inflammatory process going on of some kind, or there's B vitamin deficiencies, B6, folate, B12, etc., but that you're probably going to have to investigate more and see what's going on perhaps with that person's gut or with that person's toxin-related levels or those kinds of things. So again, this whole methylation process, if it's gunked up and not working, we have to think about neurotransmitters, DNA, think about hormone production, think about cell membranes, right? Choline and the whole connection there. Think about energy production in the muscle tissue. All these things have to be considered. And then also thinking about oxidative stress and where that's coming from in mitochondria. So it's not an isolated test that really works well all by itself, but in conjunction with these other tests that we do, this can be a really powerful tool, right? And let's see, we got a couple other examples of this test here. Yeah, oops, sorry, it's the wrong button there. And then I'll tell you what in terms of typical programs I end up designing, just to keep things easy and sort of efficient, is most of the companies that we work with have a single supplement that's designed for lowering homocysteine. I think all the major brands do. And so rather than giving a whole series of separate B vitamins, I'll often use if homocysteine is elevated, I'll often use a homocysteine lowering supplement, and it'll contain folate, B6, and B12 altogether. And then typically I'll also want to do a sulfur contain, well, depending on the labs. Let's pretend that we have a glutathione test here. So if you see a low glutathione, right, along with the homocysteine problem, then you'd want to use either the N-acetylcysteine at 3,000 to 6,000 milligrams a day, or N-acetylcysteine with additional glycine. So it's another way to do it. Practitioners sometimes will use 3,000 NAC plus 3,000 glycine. Out of the things I've learned in the last 4 years, I think the multiple roles of glycine has probably been one of the most important. Glycine, not only to help with things like what we're talking about here in terms of glutathione production, but also glycine to help with everything from gallbladder function to hemoglobin production. It's a pretty versatile amino acid. It can make a really big difference for people, and obviously a lot to do with detoxification pathways also. And the same basic story, for most of my career, underdosing people on glycine, not really understanding to start at 3,000 milligrams a day, and some people need to go up from there. If you're not getting a clinical response after a month or two, don't be afraid to bring it up. And if you are afraid to bring it up, that's okay. Just stop and retest. Do a program for 90 days and then stop and grab another test. And if you still see these problems persisting, you have the confidence to bring the dosages up. And the same thing with homocysteine. I had a patient a couple of weeks ago who had a homocysteine in the 30s. And instead of completely freaking out and panicking, I just put her on a really strong dose of B vitamins and doubled up the dose of everything and said, let's just do this for 90 days and run another lab and see what it looks like. So don't put people on high dose programs, for years or forever, but 90 days, is reasonable, maybe 6 months at the longest, and then run another test and see where you're at in terms of being able to, calm things down and start to improve things, okay? And so again, I don't think I have any examples of the glutathione labs here, but the basic, it's pretty straightforward, right? If glutathione levels are low, you use NAC, and you can combine that if you want to make it a little stronger with glycine. When the dosages start to get a little higher, it's a lot of pills. So like most of the time, the single capsules are 500 milligrams, right? So if you're talking about, let's say, I'm just making this up for average patients, say 4,500 milligrams of NAC and 4,500 milligrams of glycine, I mean, that's like 18 pills, 18 capsules, just right there. We haven't even gotten into like their homocysteine lowering stuff and all that. So I've been much more than ever before recently going to powders, okay? And there are companies that have a powdered form of NAC and glycine together, combined. And you can always get glycine as a separate product in a powder form. maybe combine that with magnesium in a powder or vitamin C in a powder. They have a bunch of ways to do it. But for a lot of these more complicated programs where I'm using higher dosages of amino acids, I can end up getting maybe 80% of the products into a powder form, which is really convenient because then the patient just does a drink in the morning and a drink at night. And then the only things that they may need to take in capsule form are things like the B vitamins or, I don't know, some of the products they may taste really horribly, just they taste so bad that people don't want to mix them into the powder, or maybe a multivitamin, those kinds of things. So there's ways you can simplify this to make it very doable. And most people can handle a drink in the morning, a drink at night, 2 or 3 capsules, breakfast, lunch, and dinner. Again, getting into these higher dosage ranges, not too many people are going to want to wrestle with, 18 capsules of glycine and N-acetylcysteine. It's just not practical for most people to be able to do that. But I'm getting great results in the last couple of years doing this, and I really want to encourage you guys to get out there and start to do these tests and see what you can get done. Okay. So I just mentioned one more time before we go to questions, we got a boot camp coming up in August. If you're interested, you get 20% off, and that's all about building your practice. We're calling it the Telehealth Business Essentials Boot Camp, but it's really about any kind of practice. I happen to work mostly on the phone with patients myself, but if you have an in-clinic practice, you're seeing people in person, it's all the same. We're looking at business planning, financial planning, strategy, basically how to set up and run the business and operations part, which is usually the least favorite thing for people to do. So going to force you, if you take this class, at least it'll force you to deal with your business for a couple of months and, get a little increase on your profitability and a little decrease on your, liability side of things and make your life a little more relaxed, hopefully. Okay, so let's open it up and see if there's any questions for folks.